Today we are going to talk about chapter 3, the molecules of cells, which are we're going to talk about the 4 classes of organic molecules. What does it mean for a compound to be organic, or what are organic compounds? Basically, they are molecules or compounds that has the element carbon in it. And then from chapter 2, we learned that carbon is very versatile, is very universal. It can form complex molecules because it has 4 valence electrons. And then these valence electrons, they'll form a bond with other molecules or atoms. So, it can bond to another atom here, another, and another. Another term to be familiar with are hydrocarbon chains. So here we have a great example of hydrocarbon chain. So hydrogen, and then carbon. These chains are mostly consisting of hydrogens and carbon, the element. And then we also call these hydrocarbon chains. This is an example of a saturated fatty acid with a long hydrocarbon chain. And then this one is unsaturated fatty acid, which has a long hydrocarbon chain as well. And then we're going to differentiate between saturated and unsaturated fatty acids later on in this chapter. Well, you see that these are all organic compounds. And then if it doesn't have a carbon in it, then it's most likely inorganic. And then oxygen, you'll usually see an oxygen in these compounds. Let's talk about isomers. So isomers are compounds or molecules that have the same molecular formula or same chemical formula, but they are shaped differently, or their bonds are different. So a great example would be 1-butane and 2-butene. Here we can see that each molecule has 4 carbons in it, and each molecule has 8 carbons in it. So they have a chemical formula of C4H8. However, we can see that their structure or shape is a little differently because of this double bond. So here, this double bond is placed at the first carbon. That's why it's called 1-butene. And then here, we see that the double bond is at the second carbon, 2-butene. So because of this double bond placement difference, it's going to make these 2 molecules different, give them different characteristics, different properties. Let's look at this example here. So butane and isobutane, they both have a chemical formula of C4H10, and then we can really tell that the structure of these molecules are different, whereas here is a long chain, okay, and then here is the carbons are branched. Well, sugars can have different structural arrangements, and then they can be isomers as well. So all these 3 different sugars, glucose, fructose, galactose, they all have this molecular formula of C6H12O6, a chemical formula that you guys will really get to know throughout the semester. Okay, but however, their structures are different. Glucose and galactose, they are both 6-ring structures, whereas here, this is a 5-ring structure. So this one's a pentose, while these 2 are hectose. And then it's because these bond arrangements are different and the shapes are different, the structures are different, it gives glucose a different taste from fructose, also fructose a different taste from galactose. Fructose is a lot sweeter than glucose. Another terminology to get familiar with are macromolecules. So macro means big, okay, and then in biology we have these big molecules that are formed from small molecules, and then these subunits, these smaller molecules, are called monomers. What we do is that we bond these monomers or put these monomers together and form a polymer. So mono means one and poly means many. these monomers, they can be identical to each other. Okay, so these subunits can be identical to each other, or these monomers, subunits, can be similar to each other. They don't have to be exactly the same. And a really good example of where monomers aren't exactly the same are amino acids. Okay, so you have 20 different amino acids, and then these 20 different amino acids come together in a particular sequence to form a polymer called a polypeptide, and then these polypeptides, they come together to form a protein. So for this exam, for this class, you guys will need to know what are the 4 classes of organic molecules, what are their polymers, and also what are their monomers. And then here, this is a really good chart, but you can find this on the internet. I believe your book has a good chart as well. So for carbohydrates, okay, carbohydrates, they form these polymers. An example of it is starch. And then starch is made up of monosaccharides. So mono means one, saccharide means sugar. So that means the subunits of starches are monosaccharides. And another word for monosaccharides we often use in biology are simple sugars. Here, nucleic acid, okay, we know that examples of nucleic acids are DNA strands and RNA strands. Well, what are their monomers? What are they made up of? They're made up of nucleotides. We string these nucleotides together to make a DNA strand. And then, of course, your DNA strand condenses to form a chromosome. Here, an example, we have an intermediate filament that's made up of several polypeptides, okay? And then remember, polypeptides are these amino acids that are strung together in a particular sequence to make this particular protein. So the last class of organic molecule, and then some books will say that it's a macromolecule, some books will say it's not a macromolecule, are adipose cells or fat droplets. Here we have these fats that can be made up of triglycerides. So triglycerides, they actually have 2 subunits. We have a fatty acid and something that's called a glycerol, which I'll explain later as well. So how do we make these polymers? How do we string these monomers together to make a polymer, to make these large molecules? Or how do we break these large molecules? How do we break these polymers into their subunits, into their monomers? We have 2 certain types of chemical reactions. First, we have a dehydration synthesis reaction. And then in this example, what we want to do is we have these 2 monosaccharides, these 2 monomers, glucose and fructose. Well, what we're going to do is that we are going to remove a water molecule. We're going to dehydrate it, remove a water molecule, and when we do that, we will link these 2, the glucose and the fructose, together so that we can form a new compound called sucrose. Sucrose. So that's formed from dehydration synthesis. Now, what if I wanted to break up this sucrose into its original monomers, glucose and fructose? What reaction would I use? I would use hydrolysis. So lysis means to break or burst, and that's one way to remember it. What we're going to do in the hydrolysis reaction in order to break this, well, we're going to add back that water molecule so that we can break that sucrose back into glucose and fructose. On page 47 of your textbook, you have a chart that is a list of chemical groups. And these chemical groups are specifically called functional groups because they will have the compound be reactive. Okay, it'll determine, how the compound will behave, or, what properties it will have. And then all of these right here are polar. So these are polar functional groups, okay, because it has the oxygen or it has the nitrogen. So remember from chapter 2, we talked about how nitrogen and oxygen really loves electrons. They are greedy for electrons. So whenever they're around, what happens is those electrons are going to spend more time with the nitrogen, with the oxygen. So that's why it will be polar. And then when we think of polar, we are going to associate it with the word hydrophilic. So hydro, water, philic means to love. That means that these polar compounds will be able to dissolve in water. They love water. On your exam, you're going to have to identify, OH. What functional group is that? Well, it's a hydroxyl group. Okay. Hydro, hydrogen, oxyl, oxygen. Okay. So here we have a hydroxyl group. And then this carbonyl group, I've X'd it out so you don't have to worry about looking at it. But of course, knowledge is power. Your next functional group that will determine the behavior of the compound is called a carboxyl group. Okay, so another name for a carboxyl group is carboxylic acid, and then it'll contain a carbon double bonded to the oxygen, and then this same carbon has this hydroxyl group on it, this OH. So basically it's COOH. So if we give you a compound structure and then we circle this part, well, that would be a carboxyl group or carboxylic acid. Your next polar functional group is called an amino group. And then this amino group will have a nitrogen and 2 hydrogens. So it'll have an amine in it. And this is also polar. Your next polar compound is called a phosphate group, and then this compound here that's shown is adenosine triphosphate, which is ATP, and then here we have a phosphate group, and a phosphate group. It has 3 phosphate groups. And then the phosphate is just, it has that phosphorus and then 4 oxygens. So all of these will be able to dissolve in water because they're polar. Remember that phrase, like dissolves like. Your last functional group, which is different from the others, so this last functional group, which is called the methyl group, it is non-polar, okay, because of the hydrocarbon chain. This methyl group is made up of a carbon and 3 hydrogens, so CH3. Because it's non-polar, it is hydrophobic. It fears water. Phobic, you have fear of something. And since it fears water, is hydrophobic, it can't be dissolved in water. So if you remember that this one is non-polar, you'll remember the others as being polar. These functional groups, we can see that you can have different functional groups in the same compound. A really good example are amino acids. So amino acids, a typical amino acids will have all of this the same, and it'll have a different R group, which is a residue group or residual group or variant group. So here we have an amino group. And then here we have a carboxylic acid or a carboxyl group. So hence the name amino acid. And steroids, really good example of steroids are testosterone and estrogen. Here we see there's an OH on testosterone, which is a hydroxyl group, a methyl group, methyl, carbonyl, which you don't have to know, hydroxyl, methyl, hydroxyl. And then coming back to amino acids. So amino acids, they can be classified as polar or non-polar, hydrophilic or hydrophobic. Here, this serine, this amino acid, is classified as polar because of this hydroxyl group, this OH. This aspartic acid is polar because it has this carboxyl group, these oxygens. And then here, leucine is considered nonpolar because of this hydrocarbon chain. So here we see two methyl groups. Oh, review question. The circle functional group in this molecule is called, so we are identifying this one. It would be a phosphate, because it has this phosphorus element and then 4 oxygens. If we were to further dissect this compound, we can see that we have 2 hydroxyl groups and an amino group. Okay. For this class, you guys will have to know the 4 different classes of carbon-based or organic compounds, the classes, and what are examples of each of them, what are the monomers of each of them, okay? So let's talk about carbohydrates. Anything that ends in OSE with the suffix OSE is a sugar. For example, we have glucose, it ends in OSE, fructose ends in OSE, galactose, OSE, sucrose, OSE, okay? And then these carbohydrates, their monomers or their subunits are called monosaccharides. Another way of saying monosaccharides are simple sugars. And then these will usually have a 1 to 2 to 1 ratio. It means that it'll have 1 carbon, 2 hydrogens, and 1 oxygen. So factors of 1, factors of 2, factors of 1. So here, can you see how this chemical formula for glucose is a 1 to 2 to 1 ratio? And we've already stated that these simple sugars are isomers of each other because they have the same chemical formula. So we have already talked about how glucose plus a fructose will make sucrose, which is just honey. 2 glucose monomers together will make maltose. And then galactose with a glucose will make a lactose. And then disaccharides are just 2 sugars together. So sucrose is a disaccharide, maltose is a disaccharide, lactose is a disaccharide. Di means 2, saccharide means sugar. Polysaccharides. A lot of times polysaccharides are made up of glucose monomers. Remember, poly means more than 1. And then there's 3 examples of polysaccharides in our everyday life. So starch, which function as an energy storage for plants, and we can find starches in potatoes. So every time we eat a potato, we're eating the energy storage that the plants will go to when they need energy. And then what reaction will they use to break up these glucose monomers in starch? They'll use hydrolysis. And what reaction will they use to link these glucose monomers together to make starch? That would be dehydration synthesis. Another example of polysaccharides that you can find in our bodies, specifically in our liver and our skeletal muscle cells, is called glycogen. Glyco means sugar. And then we make glycogen so that when we need energy, especially, for example, if we're exercising and we need some more energy to keep going, we'll go to our energy reserve in the form of glycogen, break down these glucose monomers using hydrolysis, and then our cells, our skeletal muscle cells, will take these glucose monomers, undergo cellular respiration to make ATP, energy, so that we can keep exercising. And then the last example of polysaccharide is called cellulose. Now, cellulose is the structural component for the cell wall for plants. So the long fibers of the plants that you see, especially, a good example would be on celery, are cellulose. So that was carbohydrates. Now we're going to move on to lipids. So lipids, they have these long hydrocarbon chains, okay, made up of carbons and hydrogen. And then these will usually have a ratio of 1 to 2. So 1 carbon, 2 hydrogens. And we can see that. So 1 carbon, 2 hydrogens. Okay. And then we know that fats is a lipid, and lipids are nonpolar. Okay, so these nonpolar compounds cannot dissolve in water, so it makes them hydrophobic. And then, so the fatty acid is the nonpolar part. And then here we have the glycerol head, which is actually polar. Okay, and then these come together by dehydration synthesis to form a fat. And another example of fat is our phospholipids. So phospholipids, you have 2 layers of phospholipids called the phospholipid bilayer that makes up your cell membrane. And then another example of lipids are steroids. Cholesterol is an example of a steroid, and from steroids, we can make our sex hormones, our testosterone and our estrogen. Steroids are just these 4-ring, larger structures, larger compounds. And then the last type of lipids are waxes. We've heard of beeswax, but if you look around, in nature, there's wax everywhere. Some fruit seeds will have a wax coating on it to protect it from dehydration. Even the fleshy outside will have a waxy layer on it to protect it from dehydration. Some leaves will have a waxy layer on it to protect it as well. Okay, so next time you go outside, feel a leaf. You'll feel that it's a little waxy. And then if you eat fruits, try to take a look at some of the seeds or the pits. It'll have a waxy layer on it as well. Okay, fats. So there's 2 types of fatty acids. We have saturated fatty acids and unsaturated fatty acids. So in today's society, I'm sure you guys have heard of saturated fats and unsaturated fats. Okay, well, let's take a look at what the difference is and what the whole controversy is about. So here, this picture is of a triglyceride. Okay, so this tri means 3, so it means that it has 3 fatty acid tails, 2 of them which are saturated fatty acids. And saturated fatty acid just means that all of its carbons here on this hydrocarbon chain is a carbon to carbon single bond. It means that these 2 carbons are sharing only 2 electrons. And then when you have these single bonds, it makes these fatty acid tails straight. And when you have straight fatty acid tails, and you put them together, you're able to pack them in tightly. And then a really good example of it is butter. Have you ever noticed that you can leave a stick of butter out at room temperature and it'll still be a solid? Well, it's because of these saturated fatty acid tails that are straight. They are saturated or filled with hydrogens. And then for this reason, it's harder to break down foods that have saturated fatty acids. Another good example is animal fat. So animal fats have saturated fats. For those who eat steak, you grill the steak or you put it in the pan and then you take it off, but you have to let it sit for 10-15 minutes. Well, when it's sitting there for 10-15 minutes or even longer, what happens to that fat, to that animal fat? It solidifies. It solidifies because it's made up of these saturated fatty acids. The other type of fatty acid are called unsaturated fatty acids. So these unsaturated fatty acids are a little bit better for you. They're unsaturated because they have either one double bond or more. You'll see the word polyunsaturated fats. Well, those will have more than one double bond. The carbon to carbon double bond, it just means that they're sharing four electrons, two one line and then two another line. So what happens when this fatty acid tail has a double bond is that it forms kind of like a kink in the fatty acid tail. So that means that these fatty acid tails are no longer straight. And then if it has kink and you put these unsaturated fats together that have kinks, you can't condense it. So that's for this reason, these unsaturated fats are liquids at room temperature. Liquids at room temperature. A good example is your olive oil, salmon, your omega-3 oils. Those will stay liquids at room temperature. A very important type of fat called phospholipids. These phospholipids make up your cell membrane, and then there's two layers of it. And then the way that they're oriented is unique. So here we have a polar head with glycerol, and then two fatty acid tails, okay, which are nonpolar, which are hydrophobic. So imagine if this goes all the way around a cell. On the outside of the cell, you have water. On the inside of the cell, you have water. Well, which part of the phospholipid is polar, is hydrophilic? It is the polar head that's going to be oriented towards the water on the outside and then towards the water on the inside because it loves water. What's going to be shielded from water, from both water, are the hydrophobic tails that are non-polar. So they are going to point inwards towards each other. The last type of lipids we're going to talk about are steroids. And then an example of a steroid is cholesterol. And then we've seen cholesterol. We know what cholesterol is. But we do need cholesterol because we can make our sex hormones from cholesterol, which include our testosterone and our estrogen. But we need cholesterol also because it's inserted into our cell membrane. It's randomly inserted so that it can stabilize the fluidity of it. We don't want our cell membrane to be too rigid, or we don't want our cell membrane to be too fluid, too flexible. So this cholesterol will help stabilize that fluidity. Our next type of organic molecules are proteins. So the monomers of proteins are amino acids, okay? And then this is what a typical amino acid would look like. Here we have a central carbon or alpha carbon. It has a hydrogen bonded to it. What is this functional group called? Amino group. And then what is this functional group called? Carboxyl group or carboxylic acid. So in a typical amino acid, these parts will stay the same. The only difference is this R group. It can also be called a residue group, a variant group. There's 20 different R groups, so that means there's 20 different amino acids. And then these 20 different amino acids can come in a particular order to make a polypeptide. And then these polypeptides fold together to make a protein. So let's look at this reaction. So how do we link two amino acids together? Well, we are going to take out a water, and then that reaction is called dehydration synthesis. And then when we take out that water, we are going to make a bond between these two amino acids, and then that bond is called a peptide bond. Peptide bond. So I want you to associate peptide with amino acids and then amino acids with proteins. So that's the typical structure of an amino acid. Let's take a look at the functions of proteins. Most enzymes are proteins, and enzymes, they have a very important function in your body. There's thousands of different types of enzymes. A really good example are digestive enzymes. So what these enzymes do is that they catalyze or they speed up chemical reactions. They don't particularly start chemical reactions because chemical reactions will occur anyways. It just increases the speed or the rate of that chemical reaction. In your stomach, you have digestive enzymes that will break down food. Okay. And then here you have proteins that are antibodies that will help with your immune system, that will detect a virus or a bacteria, identify them, and then destroy them. Next, we have the function of storage of amino acids. In eggs, we have the white part, which represent the ovoalbumin, and then that's amino acids for the developing embryo chick. Another function is that proteins can act as transports. So here in this picture, we have our cell membrane. Okay, two layers of phospholipids. We have the polar heads and then the nonpolar tails. Well, in chapter 2, we've already talked about how nonpolar substances or molecules like atmospheric oxygen can go in, or nonpolar carbon dioxide can easily go out of the cell without any help. However, if the substance is polar, that means it needs some kind of mechanism to go into the cell or even out of the cell. So imagine if this was glucose. Well, glucose is polar. We can dissolve it in water. It's too big and it's polar, so it needs some kind of transport protein to help it go into the cell. Another good example of transport proteins are hemoglobin molecules, which can be found on red blood cells. So what is the function of red blood cells? Red blood cells are there to transport oxygen. Well, how are they going to transport? It's the hemoglobin on the red blood cells that will bind to the oxygen and carry it into the blood vessel and carry it to your tissues so that your cells can pick up the oxygen, undergo cellular respiration so that you can make ATP. 4 more protein functions. Some hormones are protein-based. A really good example is insulin. So when you eat something, you're basically intaking glucose. But how are your cells going to know that you have glucose in the bloodstream so that it can undergo cellular respiration and make ATP? Well, your pancreatic cells, what they do is that once you have glucose in the bloodstream, in order to make it go back to homeostasis, make your blood sugar level lower, your pancreatic cells will make insulin, release it, and then this insulin will tell your other cells, hey, there's glucose in the bloodstream. Let's take it in, use it for cellular respiration so that we can undergo cellular respiration and make ATP so that we can have energy, so that we can do whatever we need to do to stay alive. And then the insulin example goes hand in hand with receptor proteins. So on your cells, you have these receptor proteins that will recognize insulin. Or another example is that you have some cells in your body that have receptors to neurotransmitters. Neurotransmitters can be released from a neuron to another neuron, or from a neuron to a muscle cell. This is how cells communicate with each other through chemicals and receptor proteins. Speaking of muscles, you have two types of proteins in your muscle cells that will allow you to contract. Okay, and then these are called actin and myosin. And what these contractile or motor proteins do is that they will slide past each other so that your muscles can shorten and you can contract. So the last function of proteins is that it can act as a structural component. Keratin, your hair is made of keratin. It has to be strong, so it's made of these proteins. And then also collagen that is underneath your skin in your connective tissue. Those are for support. Those are for structure. So before your protein can become a particular shape, it has to go through these 4 levels of structure. The first level is called the primary structure. And then this primary structure is just these amino acids coming together in a certain sequence to code for a certain polypeptide to make a certain protein. And then it depends on the sequence, what the sequence is, it'll determine the shape, the final shape of the protein. And then how do we get these amino acids together? Is through the dehydration synthesis reaction. And then what bond is used to link these amino acids together? They are peptide bonds. The next level is the secondary structure, and then this one is just how these polypeptides form patterns. There's 2 different patterns that they can form. They can form what's called an alpha helix, and this is just when it twists or coils together. Or they can form beta pleated sheets, which it kind of just folds together. There's little creases. Both of these are they have the reactions of hydrogen bonds. And then some proteins will consist only of alpha helix. Some proteins will only consist of beta pleated sheets, or they can consist of a mixture of alpha helix and beta pleated sheets. It depends on which protein it is, what the amino acid sequence is. The next level is called a tertiary structure, and this is just when it comes together to form an overall 3D shape. And this happens when the R groups, those residue groups, they come together and they can form different types of bonds. You can form hydrogen bonds, you can form disulfide bridges, which is a bond between 2 sulfur atoms. Or you can have ionic bonds, okay, so you can have a cation and then you can have an anion being attracted to each other. Or you can even have hydrophobic interactions occurring inside of the polypeptide, inside of the protein that's shielded away from water. And then the last structure is called the quaternary structure. And then this is just when you have more than one polypeptide coming together, each acting as a unit or acting as a subunit that comes together to reinforce that protein to make it strong. A really good example is collagen. Collagen has 3 polypeptides. So here's one polypeptide, here's another polypeptide, and then here's another polypeptide. So these come together to give it strength, but yet it's flexible. Another good example is hemoglobin. Hemoglobin has four polypeptides. So 1, 2, 3, 4. And then each of these hemoglobin molecules, they can bind to one atmospheric oxygen molecule. So that makes it more efficient, makes it more strong, okay? So in biology, we say shape is function. If a protein has a particular shape, then it can perform a particular function. But if that shape is somehow lost, so if it's lost, that means that that protein can no longer perform that function. We say that that protein has been denatured. If it has lost its shape, it's been denatured and it can no longer perform that function. And then there's some environmental factors that can cause proteins to denature. We can have a change in pH. Take, for example, digestive enzymes in your stomach. The optimal pH would be a very acidic pH, maybe a pH of 3 or 4. Okay, if you take those digestive enzymes away from that optimal pH and put it in a basic pH, it will no longer function. It will no longer function. Or if you add heat, high heat, or if you increase the temperature of that protein, it will no longer function as well. And then another environmental factor is if you increase the salt concentration, which is the salinity. So the last type of class of organic molecules we're going to talk about are nucleic acids, which are DNA and RNA. So we've talked about DNA in chapter 1 some, but let's really get into it. So DNA, one molecule of DNA consists of 2 strands, right here and right here, and then they are coiled together or kind of twisted, and then that's called a double helix. And the way that these 2 strands are connected to each other are through hydrogen bonds. Now, we've already established that hydrogen bonds are weak bonds, and then we choose to use hydrogen bonds here. Well, we don't choose, it just happens, is the hydrogen bonds are easy to break. And then that's good because when the cell is replicating, it needs to replicate the DNA as well. And then if we can break up these hydrogen bonds, we can make the complementary DNA strand. And then when we're finished with DNA replication, we can just reform those hydrogen bonds here. So each strand has something that's called a sugar phosphate backbone, which means that it has a sugar, phosphate, sugar, phosphate, sugar, phosphate. It's just a alternating sugar phosphate strand. And then sticking out, we have what's called nitrogenous bases. So here's one nucleotide covalently bonded to another nucleotide that's covalently bonded to another nucleotide. And we want these to be covalent bonds because they are strong bonds. We want our DNA to stay intact. So here's the typical structure of a DNA nucleotide. We have our phosphate group, we have our sugar. Okay, so in all nucleotides, it will be the same. These two will be the same. The only thing that changes in DNA is the nitrogenous base. So for DNA, we can have adenine, guanine, cytosine, and thymine. So thymine is only found in DNA. Adenine will base pair with thymine. Guanine will base pair with cytosine. And we can see that here. So here's adenine base pairing with thymine. Here's guanine base pairing with cytosine. And then this picture is just another representation. If you untwist the double helix, it'll kind of form a ladder where you have the sugar phosphate backbone on both sides. And then sticking in, we have the nitrogenous bases bonding together through hydrogen bond. For this class, you guys will have to compare and contrast DNA to RNA, okay? So one big difference between DNA and RNA is the sugar. In DNA, we have what's called a deoxyribose sugar. It just means that it's missing an oxygen at the second carbon. So here's the first carbon, here's the second carbon. It's missing an oxygen. So deoxy means that without oxygen. And then in RNA, its sugar is called ribose sugar. Ribose sugar. And then this ribose sugar does have the oxygen at the second carbon. So first carbon, second carbon, it has an oxygen. So that means it has a hydroxyl group at the second carbon. So it makes it a ribose sugar. And then here we have classification on the different nitrogenous bases. If it is guanine or adenine, which are these double ring structures, these double ring nitrogenous bases, they are called purines. And a good way to remember what purines include is pure as gold. Pure as gold. Purine, adenine, guanine. And then the others are single ring structures. These are your pyrimidines. They will include cytosine, thymine, and then when you think of thymine, think of uracil in RNA. And then one way to remember which ones are included in pyrimidine is that pyrimidine has a Y in it, cytosine has a Y in it, thymine has a Y in it, and when you think of thymine, think of uracil. Here, this is more structurally to see what the similarities are, what the differences are. So your DNA, we've established that it is two-stranded, so it's double-stranded. One strand here, one strand here. And then your RNA is single-stranded. All of your RNAs, you have 3 different types of RNA. They are all single-stranded. They both have a sugar phosphate backbone. And then they both have nitrogenous bases. So that means that they both will have nucleotides as their monomers. They both have nucleotides as their monomers. So RNA is made up of nucleotides. DNA is made up of nucleotides. DNA is an example of a nucleic acid. RNA is an example of nucleic acid. Both nucleic acids will have cytosine. Both will have guanine. Both will have adenine. It's only DNA that will have thymine. It's only RNA that will have uracil.